This is a University of Otago podcast.
Tēnā koutou, tēnā koutou, tēnā koutou katoa. My name's Helen Nicolson, the Deputy Vice-Chancellor External Engagement, and it's my great pleasure to welcome you all to this occasion of Colin Brown's inaugural professorial lecture. I'd like to extend a warm welcome to all friends, and particularly Colin's family, his wife and two boys, Neil and Alex. And we're looking forward to an exciting lecture. Being promoted to professor within the University of Otago is no mean feat. In order to be promoted, an individual has to show sustained outstanding competence in teaching, in research, and also in service. Added to that, they have to show outstanding leadership in two of the three areas. Colin is an accomplished teacher and has been Deputy Head Research in the Department of Physiology for over 2 years. He's also, as you shall hear, a strong researcher. As one of his external referees said during the promotions process, Colin is a leading international figure in neuroendocrinology and a much liked and admired member of our research community. As a previous Dean of the OSMS, I've taken an interest in Colin's progress since he arrived in 2005, both within the university and the consequences of his escapades on the soccer field. As someone whose research career began with an honours project looking at oxytocin in the brain, I'm looking forward to hearing Colin's lecture. But before we give Colin a chance to speak, I'd like to hand over to Professor Vernon Ward, the current Dean of the School of Medical Sciences, to give us some background on Colin.
Thank you, Helen. Tēnā koutou katoa, me nui tēnā kia koutou. Welcome everybody. I have the very distinct pleasure of being able to introduce Colin for his inaugural professorial lecture. Now, I think most of you know Colin, but for those of you who don't, you will work out very, very quickly that he is a Scotsman. And probably comes as no surprise then that if you look at Colin's background, he got his Bachelor of Science with honours from the University of Glasgow, and I'm going to give you the date, it was 1988. And then he followed that up by receiving a university scholarship again at the University of Glasgow, where he took on a PhD in neuroendocrinology. So he's been working in this field for some time. He received his PhD in 1993 before moving on, spending another 12 years as a research fellow and undertaking research in Edinburgh and in McGill. And before he then came, as Professor Helen Nicolson has mentioned, to the Department of Physiology here at the University of Otago in 2005. Probably is no surprise to you that he's a member of the Centre for Neuroendocrinology, and he's also a member of the Brain Health Research Centre, a couple of our real flagship centres for this university and an integral part of them. His research expertise, again, follows down his neuroendocrinology path. It's clearly neuroendocrinology, there's reproduction, neuroscience, a lot of electrophysiology, and I think you'll see all the real skills and techniques and knowledge that goes with those fields. As we already know, you have to be strong in research, along with the other skills of service and teaching in order to become a professor. This is his inaugural professorial lecture, so I'll focus a little bit on his research, which is, he's got extended research funding right now. He's a co-PI on an HRC $5 million program grant. He's a co-PI on that. He's had funding from Marsden, Lottery Health, and in the past Wellcome Trust, so he's been well funded. He has close to 70 research publications. Some of the highlights from that would be Neuron, and for those of you that like to count such things, that's an impact factor 16, which we all think is fairly good. Journal of Physiology, Neuropharmacology, Endocrinology, Journal of Neuroscience, all good, strong core journals in his discipline. As already mentioned, his service involvement is strong in and around Deputy Head for Research for the Department of Physiology. He's also on the Otago PBRS Steering Committee, which is quite an important role. His list of memberships of societies is long. His contribution to them is extensive, and he's a senior editor for the Journal of Neuroendocrinology, so he's well-rounded. So his promotion is very well deserved. Now, I also have to give a little brief introduction to his talk without trying to steal his thunder. And so his talk is Oxytocin, Neurons, Keys to Unlocking Brain Function. So I asked Colin a little bit about his presentation. I think what he's looking to do here is develop a little theme along the way of today's talk. A simple quest to work out basic science of how a system actually works, and then that leads to fundamental discoveries. I think it's something many of us aspire to. Then you take those discoveries and you have the potential to have a direct impact on clinical practice. And for those of us that do research, I think that would be a wonderful outcome to know that your research had an impact out in the real world. And so what he's going to talk about today is a path, and it's going to be looking at the identification of a previously unknown mechanism that might control birth. And then this has the potential to lead on to new types of drug controlling things like preterm birth, getting better drugs, that sort of work. So I don't want to take anything away from his presentation by going on any longer. So at this point, I would like to invite Professor Brown to present his inaugural professorial lecture. Nō reira, tēnā te mihi, kia koutou katoa, tēnā koutou, tēnā koutou, tēnā tātou katoa.
So thank you for the kind introduction, Vernon. I'm not sure I recognise the person you're describing there, but we'll go with it. So as Vernon said, what I'm going to try and do tonight is take you through the journey that I've been on through my research career. So that started, as Vernon said, with a basic quest to understand one thing about how one particular brain cell works. And as that theme has gone through and been expanded as I've gone on. So where I'm going to start is just on communication in the brain. So this little squiggle represents the mechanism by which brain cells or neurons talk to each other. It's called an action potential. So this shows the voltage measured from an individual brain cell, an oxytocin neuron. Actually, it's a recording I made personally about 18 years ago. And this is the way that brain cells communicate with each other. So they send electrical impulses, and when you watch CSI and the like, and they show those little lights flashing inside the brain, this is what they're trying to represent, these electrical impulses. But what we don't know is what those impulses are actually saying. You know, what's the message they're giving? We know how they're doing it, but what's the message? That's because there are lots of things going on. Difficult to identify individual cell types to know what they're doing. And what's happening inside the brain doesn't fit very well with what we see in the real world. So this is real world communication. Anybody want to hazard a guess what I'm showing? Oh, someone's got to say something loud enough that I can hear it. Okay. So this is pretty much how I'm feeling right now. SOS, SOS, SOS. But the illustration here is actually fundamental to our understanding of how the brain functions. Morse code is just dots and dashes. So you can summarize the sum total of human knowledge with only 2 symbols. Those of you who aren't quite as old as me might recognize this kind of representation of information. Binary code from computers, zeros and ones. And again, these zeros and ones on their own don't confer any information whatsoever. What matters is how they appear in sequence with all of the other zeros and ones. So in this ASCII code, 0 is 0011 0000. So this 1 means absolutely nothing unless you can see what's happening on either side of it and put the individual word that number is representing into context with everything else around it. So now what we can see is action potentials occur in sequences. These electrical impulses, these signals that communicate from one brain cell to another brain cell, occur in sequences. Different sequences for different types of brain cell. And what I hope you can look at now is look and see that these intervals are different. And if you now translate these action potentials and think of them as, if you like, as an up state, and the baseline membrane potential as the down state, or as zeros and ones, you can start to code the information. So it's not the occurrence of a single action potential that means anything. It's what happens in that sequence. So of course, this is easy to say when you've got one cell, and you can see what's going on with that one cell, but the brain is incredibly complex. If I'm not sure I'll get Phil Sheard's quote correctly, but it's the most complex thing in the known universe. And that's where our trouble comes, is how do you decipher those messages when you don't really know what's going on? It goes in and it comes out. So this is where we apply a scientific method, reductionism. Contract the system to a manageable, a bite-sized piece of system. And this is my favorite model of brain function. It's the one that I came to neuroscience with, because I was trained in physiology, and my neuroscience training was almost zero within that physiology degree. So you have sensory information, comes in at one end, and hopefully the appropriate response comes out at the other end. And something magical and mystical happens in between. And that something magical and mystical usually involves complex networks of brain cells that communicate with each other. And this is where neuroendocrine systems come in. So for this particular set of brain cells that I'm interested in, we actually know what the input is. So for these brain cells, they respond to the baby suckling at the nipple during lactation. And the response is to deliver milk to the suckling baby to satiate the hungry child. And the bit in the middle boils down to these oxytocin cells, oxytocin neurons. So these are specialized brain cells that, like all other neurons, sit inside the brain, receive lots of inputs, but instead of making connections with other brain cells, they send their projection to a specialized gland called the posterior pituitary gland and secrete the oxytocin into the bloodstream. So the neuroendocrine, neuro because they're brain cells, and endocrine because they actually secrete a hormone rather than a transmitter within the brain. So we can understand what the purpose of the activity of these neurons is quite readily. And this now shows those action potentials firing in a different way. So this is now no longer the voltage measurements from a single cell, but the translation of those, the currents of those action potentials into a firing rate. So this is spikes per second, or hertz, on the y-axis, and the time in minutes on the x-axis. And what you can see is that most of the time, this oxytocin cell is just bumbling along, doing nothing particularly spectacular. And then once every few minutes, there's this very high frequency burst of action potentials. And that burst of action potentials releases a pulse of oxytocin into the bloodstream. And that pulse of oxytocin travels around the body and causes contraction of the muscle cells in the milk ducts, which then causes the delivery of the milk to the suckling baby. It's not the only thing that oxytocin does. It's actually also required for normal birth. And essentially, although the stimulus and the outcome are different, the processing in the brain appears to be indistinguishable. So the two things that oxytocin is doing is essentially getting your baby delivered and keeping it alive once it's delivered. So we know what's going on at this level, and actually we've known everything that I'm showing you just now was textbook physiology by the time I started my undergraduate degree in 1983. So we've known this goes on, but actually we don't know how this happens. Because if you think about it, baby suckling at the nipple is continuous stimulus. The baby latches on, suckles all of the time, and somehow the brain is taking that continuous signal and transforming it into this episodic output, these bursts of activity in these cells that cause the pulsatile secretion of the hormone. And that is processed at the level of the oxytocin cells themselves. So when we look at the activity of the brain cells and the network around them, that looks random, even when there is suckling of the baby. Oxytocin, of course, is far more important than that. I see Dave Grattan arrived late. Well done, Dave. Dave tries to persuade everybody on the planet that prolactin is the most important hormone in the world, but as you'll see later, it's only important because it's involved in the regulation of oxytocin secretion. And oxytocin is, for those of you who are more classically trained, oxytocin not only helps you give birth and keeps your baby alive, but actually it's responsible for the creation of the Milky Way where we all live. So this is Heracles suckling at Hera's nipples. And Tintoretto, if he wasn't such a great artist, could have been a physiologist, because here he's actually captured the physiology perfectly. The baby is suckling at one nipple. You might have noticed there's actually two nipples there. And the spray of milk that's making the spray of stars that is the Milky Way is coming from both nipples. And that's because the baby suckling at this nipple is sending electrical impulses to the brain of Hera, who is then secreting oxytocin in pulses that travels around the body as a hormone in the blood system and arrives at both nipples. So the milk gets ejected from both nipples in response to suckling at one. Tintoretto isn't the only person, only artist to treat this subject. Rubens had a go at it several decades later, and at first glance you might think, oh, Rubens not such a good physiologist as Tintoretto because the milk's only coming out one side. But actually, Rubens has caught it perfectly as well, because here, in this instance, Heracles isn't actually suckling at the nipple. Quite painfully looking, it's a manual squeeze of the breast that's causing the milk ejection. And that's not the appropriate stimulus for oxytocin secretion from the brain. Suckling at the nipple is. So here, the milk is only coming out one side because the stimulus is not eliciting the appropriate response to get the milk ejection from both breasts. So that's my pretense at being well-educated, and we'll come back to physiology for the rest of the lecture. So this is now essentially Human Body Systems 191. So for birth, this whole system is called the Ferguson reflex. And the start of the process is when the baby's head pushes down on the cervix. And that causes stretch of the cervix. And that stretch is sensed by special receptors in the cervix, which sends nervous impulses up to a specialized bit of the brain called the hypothalamus, which controls the secretion of most of the hormones in the body, including the secretion of oxytocin. So the oxytocin cells reside within the hypothalamus. And when you get that stretch of the cervix, you get an increase in this action potential frequency. And as I told you, this is not just a general increase, it's an episodic bursting response to that stretch. These oxytocin neurons send the projections to the posterior pituitary gland, where they secrete the oxytocin into the blood. So that increases oxytocin levels, which in this instance causes contraction of other muscle cells in the uterus to cause the delivery of the baby. So this, again, as I said, textbook physiology, even when I was still relatively young. But what I became interested in is what mechanisms drive this pattern. How do these neurons take this continuously delivered information and transform it into patterned output? Because perhaps this is the way that brain cells function. Not in the simple integration of all these inputs coming in, and here I go, passively, but here I have properties and I can process signals and make a decision about how those signals are relayed. But of course, much smarter guys than I had been dealing with this for decades. And it's quite daunting to come at these things. So I was left with this kind of thing. If you can't be as smart as everybody else, what can you do? Well, you can work pretty hard at it. So eventually, the movie industry caught up with my thinking and came up with a quote that I've been looking for my whole life. So the question then became, how am I actually going to science the shit out of this? How am I going to do something that actually makes a difference to our understanding? So we've got the baby, it's coming, and get that thing out of me. So people have been working on it and working on this particular activity pattern and really getting pretty much nowhere. So for reasons I won't go into, I took a totally different tack from the field. And that was to look at the effects of the painkiller morphine on oxytocin cells. And I can see June Kim almost getting interested now. June's the latest student to be subjected to this theme of research. So this diagram shows the firing rate of another oxytocin cell from actually from a brain slice prepared from a rat by a colleague of mine, John Wakerley. I chose this particular example because it was published in the year that I started my undergraduate studies. And what he shows here is this oxytocin cell is switched off if you apply morphine, the painkiller. And if you give a drug which blocks the actions of morphine, you can get the recovery of the activity of this oxytocin cell. But the remarkable thing about these cells is that you don't actually have to give this naloxone to block the effects of the morphine and get recovery. If you give the morphine to these brain cells for long enough, they'll start firing again. They'll change their physiological state to essentially a pathophysiological state, and they'll require the morphine to be there to function apparently normally. And when you take the morphine away at that point, if you allow them to recover first and then give the drug that blocks the morphine effects, you get a syndrome called the withdrawal syndrome, which is analogous to cold turkey addiction. So this process is called morphine dependence, and it's one of the physical processes that underpin the psychological process of addiction. But importantly, what we also knew from work that others have done is that there are drugs, or chemicals inside your brain that are the same as morphine. That's why morphine works, because it's mimicking the effects of something that's already there. And what we know is that, what we knew is that those endogenous substances, those chemicals released within the brain, actually inhibit oxytocin cells over the course of pregnancy. So when I come into this, I come in with the idea that perhaps that what happens is the oxytocin cells are kept quiet over pregnancy by these endogenous, these natural morphine-like substances. And then when you're going to give birth, that inhibition is removed and you get the excitation. So, some might say that morphine's more fun than getting pregnant. I have to admit, I've never tried that particular drug, so I can't express an opinion. But I can summarize somewhere about 5 or 6 years' work in one slide. This one slide that sadly has never been published. So here, this is a recording of an oxytocin cell from an anesthetized rat that has been given morphine for 5 days. And then it's given the drug that blocks the effects of that morphine. And what you can see is there's an excitation, and importantly, on top of that generalized excitation, you can see not identical to what you see in suckling, but bursts of activity every few minutes. And of course, I saw this and I thought, well done, we've sorted it, worked it out. But the scientists in the audience will be saying, where's the replication? And the simple fact is, the reason this is the first, to the best of my recollection, this is the first time I've ever shown this data to anyone outside the lab. We never replicated this. So we've done this experiment for different reasons in different ways, but we've done it possibly even hundreds of times, and this is the only cell that ever responded the way that we wanted. And you don't get a Nobel Prize for N equals 1. I know, I asked, they said no. So, what do? You've had your idea, still think it was a reasonable idea, but it wasn't right. So at this point in my career, so that recording should have said was, I think, somewhere about 1996 we've got to so far, so we should be finished by about 8:30. I moved on to another cell type, actually, so looking at vasopressin cells in the brain, and the reason I was chose to do that is because the vasopressin cells and the oxytocin cells are almost identical. They're different from each other, but almost twins. Vasopressin cells have got nothing to do with reproduction, at least at the level that I've been talking to you about. What they deal with is body fluid balance. So when you're thirsty because you're running out of water. Last thing you want to do if you're running out of water is go and pee what you've got left up against the wall. So what vasopressin cells do is actually act on your kidney to reabsorb the water in your urine so that you don't lose any more water. Prolonging how long you'll stay alive, but not keeping you alive unless you actually go and get more water. The reason I moved on to the vasopressin cells was because the vasopressin cells also show a bursting activity pattern. It's not the same as the oxytocin cells, but it has similarities. So here, this is the firing rate of a vasopressin cell recorded from an anesthetized rat. So again, the firing rate in spikes per second on the y-axis, time in minutes on the x-axis, and you can see these bursts are much longer and a bit not quite so high amplitude. Two advantages to looking at vasopressin cells is, one, the bursts are longer, so you've got more time to actually do something to investigate what's going on during those bursts. The second reason is that you need to control your water balance all of the time. So you don't have to work on pregnant or lactating rats and go through all of that and look for a specific phenomenon that only occurs at one specific time of life. And actually, even during that, it only goes for 2 seconds every 5 minutes when it works. So a large proportion of vasopressin cells show this behavior throughout life. So I and others in the field spent quite a long time. This is a 10-year vignette for me, trying to work out how this pattern is set up, with the idea that we might be able to use that to inform our understanding of what might be going on in oxytocin cells and apply experiments to investigate that system. So this sums up where we got to. So this, again, is a single action potential. So this is a voltage trace, a single action potential, this time from a vasopressin cell. And the inset is what I want to draw your attention to. So this is the action potential here. And what you can see is after the action potential, there's a very, very small but long-lasting excursion in the voltage. And by long-lasting, if you look at the time scale, this is several seconds long. And in brain communication, several seconds is an eternity. Communication, most people in neuroscience who think about millisecond communication. So this changes the excitability of the vasopressin cell. So what I didn't say to you earlier is these action potentials, these electrical impulses are all or none events. So if you make the voltage go far enough up, you will reach what we call threshold, and then you get your action potential. In this instance, the voltage didn't reach that threshold, and so it just slowly decayed back down to baseline and bumbled along, doing nothing very exciting. So that voltage excursion is called an after depolarization. It's a depolarization of the membrane voltage, and it happens after an action potential. It's caused by the action potential itself. But sometimes you'll get one of these action potentials with your after depolarization, marked by the little asterisk, and something else will come in and make the cell reach threshold. And you get another action potential. And because that after depolarization is so long-lasting, the second action potential is superimposed on that first depolarization. So they add up. And eventually you reach a point where you reach a new steady state where the cell is more excitable because it's closer to the point where you can trigger that all or none action potential and send a signal to, if you're a run-of-the-mill brain cell that I'm not very interested in, another brain cell in the brain, or to the posterior pituitary gland and secrete the oxytocin, or in this case, the vasopressin into the bloodstream. So I spent about 10 years working out how this works. The actual contribution I made isn't on this slide. But what we did do through work by a colleague and friend of mine, Javier Stern, while he was working in Bill Armstrong's lab, they identified that this phenomenon is not normally seen in oxytocin neurons if you record them from a virgin rat. But if you record them from a lactating rat or a late pregnant rat, you do see this phenomenon. So if this phenomenon emerges in late pregnancy and remains during lactation, you would guess that it's probably doing something to do with pregnancy and lactation. So we turn back to our oxytocin neurons with the question, does enhancement of that after depolarization that was identified, does that increase the activity in such a way that it could underpin the behavior that we see in birth and lactation? So we went back to the in vivo situation. So this, again, is another recording from an anesthetised rat. And the dependent here means that this is from a rat that was treated with morphine for 5 days again, because we decided we wanted to give ourselves the best chance possible. If it worked one out of several hundred times in the other experiments we did, maybe it would work more often if we do the experiment in these ones. So we gave a drug called apamin. It's actually the active ingredient in bee venom, so this is what makes it sting, and saw absolutely nothing. So we gave a higher dose, as you do. If at first it doesn't work, hit it with a bigger hammer. And unfortunately, that didn't do anything. So let's hit it. Now let's put our hammers down and pull a sledgehammer out. So we hit it with the naloxone, the drug that blocks the effects, the ongoing effects of the morphine, which we know causes a robust excitation. And when we do that, we see nothing, or nothing different from what we see when that apamin isn't present. So, playing around with if you like, the endogenous morphine, the natural morphine, and playing around with the intrinsic properties, the after depolarization of these cells, even when you put them together, we can't replicate that lusting behavior. And that's science the way I see it. We didn't manage to do it. And actually, with my usual level of intelligence, after 15 years of trying, I realized that I was probably wasting my time. Because birth and lactation for mammals, are absolutely essential, not only for your survival or your family survival, it's absolutely essential for survival of the species. So why, oh why, if you believe in intelligent design, would you design a system that will fall over if you poke it in one way? So actually, in reflection, it's no surprise to me that you can't replicate this behavior by changing one or two things, because the system itself, I would expect, is going to be multiply redundant. You would not go out your way to design a system that will fall and fail and be the end of your species. And if you believe in evolution, I think something that evolved randomly that way also would mean that we weren't here in this room tonight. So, that point actually coincides with just about the time that I came to Otago, even though some of the experiments were done after I arrived. And what we've done in the lab here, which I'm going to deal with in the last few science slides, is actually stop trying to work out what causes that bursting. Yeah, I've backed off from it. I've parked it in the too hard basket, and maybe another decade down the line, I might come back to it as the swan song of my career. But what we've become interested in is not what actually causes the bursting, what triggers each individual burst, but what changes the excitability of the oxytocin cells at the end of pregnancy and during lactation that will make it easier for whatever it is that is processing that signal to be successful, to actually drive the increase in activity that we see at this time. So as I said earlier, I collaborate with Dave Grattan, and Dave, funnily enough, persuaded, this is 2009 we started this, to look at prolactin effects. So the two hormones that control lactation are oxytocin, which causes the milk ejection when the baby suckles, and prolactin, named for its effects in lactation, prolactational, causes milk synthesis. But it was known that actually if you give prolactin into the brain of rats, and we've done this here again in anesthetized rats, in virgin rats, prolactin will inhibit oxytocin cells. And that's fine, because in virgin rats, they're actually doing something else. Prolactin, is it over 400? Certainly over 300. 300 different actions. The one it's named for is the one we're interested in. Oxytocin also has lots more functions, but we're not going to mention the dirty words of love hormone tonight. So here we have prolactin inhibiting oxytocin neurons. But what we found is if you do exactly the same recordings using exactly the same conditions, but in an animal that's lactating, what you find is if you give prolactin into the brain of that anesthetized lactating animal, some oxytocin cells will still be inhibited, very similar to what you see in the virgins, but not many. Most of them are actually not affected at all. And some are actually excited. So looks like there's at least a loss of the inhibition and perhaps even a switch to an excitation. So in virgin animals, you don't want oxytocin and prolactin to be secreted at the same time. But in lactating animals, and perhaps at the end of birth, and we haven't tackled that problem yet, you want prolactin and oxytocin to act in concert. You want them to reinforce each other because you want to synthesize milk at the same time as you're delivering milk to the young. You need to replenish your stores of milk. So having a system that actually does that, it seems quite intuitively obvious. And this is work, sorry, I should have said Rachel, that has been led by Rachel Augustine in the lab for the last few years, ably helped by Greg Bauer. The other thing we're looking at in the lab that might be important, and this is actually probably the most exciting thing, but I'm so Scottish you won't catch the excitement in my voice when I tell you it, is this other neuropeptide class called crispeptin. So what we've discovered through work of Xander Seymour, who just finished his PhD in the lab working on this topic, is that a specific group of crispeptin cells appears to act on oxytocin cells in the brain at the end of pregnancy. So here, this is an image of a bit of a slice through the brain of a rat where you find oxytocin cells. So the green blobs are essentially oxytocin cells in the brain. And you'll have to take my word for it at the moment that the red is crispeptin staining. There is some there. And as you go through pregnancy in the rats, there's more and more and more. So in our rat colonies, day 21 of pregnancy is the expected day of parturition, the expected day of delivery of the young. And what Xander has identified is that these cells that synthesize crispeptin, a specific subpopulation of them, increase the amount of crispeptin they synthesize towards the end of pregnancy. So if they do that at that time, again, intuition would suggest that possibly they're doing something to do with pregnancy. So Vicky Scott, who was a postdoc in the lab for a number of years before she turned to the dark side and enrolled in medicine, did the electrophysiology on this kisspeptin. So when you work on an anesthetized virgin rat, when you inject kisspeptin into the brain of that virgin rat, it has absolutely no effect. In early pregnancy, some cells respond. Not many, but some do. Mid-pregnancy, again, some cells respond, most don't. But at the end of pregnancy, in the last couple of days of pregnancy, all oxytocin cells are excited by kisspeptin injected into the brain of an anesthetized rat. So the scientists in the audience will have recognized that I've shown you absolutely no data whatsoever tonight. I've shown you a bunch of examples and asked you to trust me. So on the final science slide, I've actually put some results in. So the line that I've been feeding you is actually correct. So this just quantifies the electrophysiology results that were shown in the previous slide. So the NP is non-pregnant, change in firing rate, so 0 means it didn't change. After injection of kisspeptin that I showed in the previous slides. So in the non-pregnant animals, there's a scatter around 0. Early and mid-pregnant animals, some cells clearly respond, and that's what these grey dots are. The grey dots are the individual measures from individual cells, and the black bars are the average responses. Whoops. But in the late pregnant animals, all of the cells respond to kisspeptin. So what we think is going on, and it is still just think, this is a working hypothesis. We don't know if this is true, and if there's any funding agencies in the audience, we'd be very happy to receive more money to try and work it out, is we have these kisspeptin cells in the brain. We have our oxytocin cells in the brain and some other kinds of cells. So over the course of pregnancy, these kisspeptin cells increase the synthesis of kisspeptin. And we think, actually, they probably don't have a direct effect on the oxytocin cells themselves. We think it's probably mediated by some other neuron brain cell in the brain. What we know about this population of kisspeptin cells and the oxytocin cells themselves is they actually express the proteins that allow them to respond to prolactin, or more specifically in this case, placental lactin, which works on through exactly the same systems as the prolactin that I talked about earlier. So what we're currently investigating in the HRC program grant that Vernon mentioned in the introduction is trying to work out whether this prolactin drives the changes in these kisspeptin neurons that increase the excitability of the oxytocin neurons at the end of pregnancy. Maybe also the prolactin acting directly on the oxytocin cells to directly increase their responsiveness to these excitatory inputs. And the key thing about this, the thing that's actually most exciting about this, is so far as we're aware, this is the only thing that kisspeptin is doing in the brain at the end of pregnancy. So if we can target that particular system, then we have a potential to reduce the excitability of the brain cells that secrete the hormone that cause birth. So for individuals at risk of preterm labor, we might be able to develop drugs, a different class of drugs that will reduce the risk of preterm labor without affecting I said I wouldn't say it, but I'm going to, the love hormone side of the oxytocin equation, which is not the oxytocin that's secreted into the blood. Oxytocin, other cells secrete oxytocin into the brain, and that's all the oxytocin that's involved in social bonding. And if you give drugs that interfere with oxytocin signaling, you'll interfere with the social bonding and actually the maternal bond. So if we can isolate this population of oxytocin cells, which are the only ones that are affected by this kisspeptin input, then we have a new target for drugs to protect against preterm labor and to stop the delivery. Oh, I meant to delete this bit. So we think this population of neurons is probably expressing another transmitter in the brain called noradrenaline. So what you'll recognize is I didn't actually get to the end point of where I started, and I think that's the way that science works. You start with a goal in mind, and after 2.5 decades, in my instance, of working through all of this, still trying to science the shit out of it. Don't science the shit out of anything on your own in this game. And increasingly, science is becoming a collaborative effort. And I'd just like to acknowledge the people in the lab. Rachel, Greg, and Xander are the people whose work that I actually showed you. But we also have Aaron and Ranjan who are doing work on vasopressin cells and actually how that impacts cardiovascular function. And all of those projects are in collaboration with people at Otago. Lucky enough to have good, strong collaborations with international leaders around the world. And again, as Vernon said, none of it would be possible without the funding agencies who've kept us going, kept me going for 25 years. But even that, I think, is not sufficient because the physiology department is, I think, at this time, over 100 people, Brian? All pulling in the same direction, trying to achieve the same goals. And I get time to do all of the fun stuff, all the teaching and the research, because the physiology department, I think, functions really well as a team. And I'd just like to take this opportunity to particularly thank the general staff in the department, because all the shit that you guys do for all of the administration that idiots like me don't have to do make it much easier for us to succeed in our research as well as in our teaching. But you don't get the biggest thanks. I still haven't managed to see, oh yeah, I heard you, but I didn't see you. So sadly, I have to say, there's only one Prime Minister's Science Prize winning member of the Bruins. And it's not the big fat asshole in the orange jersey. So I've got a lot of catching up to do with the rest of the family, as do the kids. So if you didn't understand, it's Evelyn Armstrong who works in the chemistry department, who has won it and has been my partner for 25 years, and I'm looking forward to the next 25 if I can last that long. And the final thing I have is just a message to any of the younger members of the audience. Those of you who have known me reasonably well for a long time know that I am an ex-movie star. Well, movie star might be a bit of an exaggeration. I appear in the movie Trainspotting for about 1.5 seconds as bemused bystander number 2. And that's just by way of introduction, because that story goes around. And it went around to Tiffany Fleming, who was a visiting student, who came to my lab from Javier Stern's lab. And when she went back, she found this and sent it to me, and it's still my favorite image about research. I'm not going to read it all, because you can read it quicker than I can say it. But I've been in the game long enough that I have actually suffered every single thing on this list, apart from the ulcers. And I'm working on that now. But despite that, I mean, I can't think of anything better to do with your life than to create and disseminate new knowledge. And despite all of the crap that goes on and all the complaining that I and others do, I would still choose research. So with that, I just thank you for your attention.
Thanks, Colin. I'm Brian Hyland, and it's my honour to be the current Head of Department of Physiology, and I have a couple of roles to do now at the end of this presentation. First is to formally thank Colin for the presentation, and I was told that I have up to 10 minutes, so I'm pleased you sat down. I think, you know, what Colin has achieved here tonight is to give us a very nice example of how a researcher and the team, colleagues, collaborators, working away, unpicking a piece of the puzzle. The brain is this huge puzzle, and it's made up of many, many individual pieces, and there's enough work in there for many of us to work for our entire careers working on each small piece. But by addressing this wonderful, I think Colin, really showed it up in the way it is. It's this dance, this symphony of electricity and chemistry underlying one particular pathway, which in the end focuses on a single hormone, single chemical, and yet is involved in, as was discussed in the talk, over 300 separate functions. Each person is working away, unpicking that piece of the puzzle, and as part of a bigger enterprise across the university here, around the world, that's needed to address, of course, what is, as Colin pointed out, the single most complicated thing in the universe. And I think, it's a really nice example of that interplay, as I said, electricity, chemistry, dogged perseverance and hard work, which is coming together to produce a better understanding of the basic physiology, the basic processes, and through that, the opportunity, and one never knows where this is going to arise from basic research, to identify targets, identify causes of problems in the brain, and identify ways to address those problems and treat conditions which affect our health and well-being. So once again, I'd like you all to join me in congratulating Colin on ascension to professorship and for his presentation tonight. Now, the next thing I get to do is to give out the goodies. So if I can do this one-handed, Colin, you will have to stand up again. So what we have traditionally in these processes is we give the presenter, a couple of recognitions of the event, because it is a very important event in an academic's life. So Colin, here's a big thing for you to remember the event by.
It's a picture of me. Just what I always wanted.
And I have a smaller thing, which is not what you think it might be, the envelope brought out by the head of department. But no, this is a small thing, but actually quite an interesting thing, I think you'll find. So once again, Colin, thank you, and hope you enjoy the products of that envelope. Okay, so the very final thing I have to do then is to point out that Colin didn't have time to get very much into some of the other functions of oxytocin in the brain. And he did sort of intimate a little bit to what we could perhaps delicately call the pro-social aspects of oxytocin function in the brain. That is, it's very important for promoting social bonding and interaction. But it is also important it's activated by feeding and drinking, as it turns out. And luckily, we have an opportunity to test that hypothesis, because it's my pleasure now to invite you all to come and join us for the final element of the celebration, which are refreshments in the staff club. Upstairs in the staff club, you find your way, if you don't know, through the works around here. You leave the lecture theatre on that side. Come and join us to celebrate with Colin with those refreshments. And finally, it's just my great pleasure to thank you all, the audience, for coming along to this event.